Okay. Welcome to this webinar. I thank very much all of you to participate to this webinar focusing on harmonization of autoimmune tests. Just very few words to introduce today's webinar. As you know, there is an extraordinary development in autoimmune diagnostic characterized by the progressive evolution of analytical technologies, the availability of new tests, the introduction of automation and consolidation in laboratory. Antibody measurement in laboratory is an extraordinary tool to make a robust and often conclusive diagnosis of autoimmune disease. But variability in analysis and reagents, as well as techniques heterogeneity, are causes for discrepancies or disagreement, strong disagreement in interlaboratory variation and the test result report. And this lack of reliability in laboratory information may be responsible for wrong or missed diagnosis, as well as additional cost. So the importance of standardization or harmonization in laboratory medicine, and specifically in autoimmunity, is a key issue, as the affinity of antigen-antibody binding may vary between and within methods, with a consequent well-known marked variation in autoantibody concentration measured by the different analytical methods. The automated quantitative autoantibody methods implementation has made standardization or harmonization of these tests even more necessary and required to provide accurate and similar test results for an individual patient at any time and at any place. Today, Dr. Joanna Sheldon will present this challenging issue, and it is my great pleasure to introduce you Joanna Sheldon, a leading scientist in the field of laboratory medicine and autoimmunity. Dr. Joanna Sheldon is a consultant clinical scientist in immunology and director of the Sub-Regional Protein Reference Unit at St. George Hospital in London, part of South West London Pathology. The Sub-Regional Protein Reference Unit is a National Health Service laboratory serving, receiving samples sent from all around the UK, and the test repertoire covers allergy, autoimmune serology, cerebrospinal fluid analysis, cytokine, and monoclonal protein identification. Dr. Sheldon is particularly interested in training and education and in delivering high-quality results. Dr. Sheldon was part of the IFCC committee that oversaw the production of the international reference preparation for plasma proteins. Dr. Sheldon, as well, now chairs the IFCC committee on the harmonization of autoantibody tests that is currently working on preparation and validation of international reference materials for autoantibody. Please, Joanna.
Thank you so much, Tommaso, and thank you all very much for being here in your evenings to learn a little bit about harmonization of autoantibody tests. I'd like to thank the EFLM for their foresight in choosing this as a topic for one of these sessions, because it's a real challenge for us in laboratories. Now, this is the very first webinar I have ever done, so you have to bear with me if I'm a little bit slow in figuring out what I have to do. But just to give you a little bit of perspective, really a lot of the work that I'm going to present today is a result of the work that was started by the International Federation of Clinical Chemistry and Laboratory Medicine, the IFCC, and about 7 years ago they formed a working group which subsequently became a committee for the harmonization of autoantibody testing. And of course none of the things that I will show you today are just me. In fact, I only play a little part in them. We have many, many people to thank, the people who have prepared these materials, evaluated them, given us very sound advice, the patients who've given their materials for developing these reference preparations, and all the many people who've been involved in this project. So thank you as a beginning to all of them. But if we're going to talk about autoantibodies, we have to think a little bit about autoimmune diseases. As I'm sure many of you know, these are a diverse group of diseases that can affect anything from one organ of the body through to many organs of the body, or being entirely systemic or non-organ specific autoimmune diseases. In addition to them being a diverse group of diseases from the parts of the body that they affect, there's also a spectrum of severity. You can have patients with diseases with symptoms that are easily treatable. For example, if a patient has well-controlled rheumatoid arthritis, and right through to the other end of the spectrum when the patient has a life-limiting condition, for example, systemic lupus erythematosus or an ANCA-associated vasculitis. Now, we as healthcare professionals, we can sit in our offices with lots of books on the wall and experience and colleagues who we can discuss. But if a patient was all of a sudden diagnosed, for example, with SLE, they would probably type that into a computer and say, oh my goodness, what's SLE? What does it mean? And if we look at SLE from Wikipedia, we see it can be fatal. And these are the kind of words that would jump out at the patient. There is no cure. It can affect the heart and the lungs and the skin and the blood vessels, the kidney, liver. So really, when a patient gets the perspective without a sensible or accomplished clinician helping them, they can be very scared of the condition that they have been diagnosed with. Similarly, if we go to the doctor internet, somewhere on the internet, and we're looking at ANCA-associated vasculitis, we have very, very serious symptoms, rapidly progressing renal failure, for example, that a patient would be very, very scared if they had that diagnosis. Now, of course, if they had the condition and we made the correct diagnosis, that would mean the patient was on the correct path to treatment. But if we, for example, made an incorrect diagnosis, we didn't diagnose a patient with an ANCA-vasculitis when they had one, or we falsely gave that diagnosis to a patient who didn't have the ANCA-vasculitis, then the consequences, of course, are very serious. Particularly because some of the drugs that are used in these conditions are not just a little bit of paracetamol for a headache. These are heavy-duty immunosuppression, high-dose steroids, cyclophosphamide, and plasmapheresis are some of the drugs that may be used in managing patients with autoimmune diseases. And in the laboratory, we don't see the patients directly, but we often get a blood sample, and in that blood sample, they're asking for tests. They're asking for us to detect or quantify the amount of autoantibodies in that patient sample. And an autoantibody, again, as I'm sure you all know, is an antibody, often an IgG antibody, to an antigen that's expressed in our normal tissues. And there are many examples of autoantibodies. For example, thyroid peroxidase antibodies are one of the antibodies that were very familiar to be used in investigation of patients with hypothyroidism. Antibodies to proteinase 3 or myeloperoxidase, these are enzymes in the neutrophil cytoplasm, and they're important in looking at patients with ANCA-vasculitis. But actually, everybody has autoantibodies. If we took blood from all of the participants in this webinar, we would all be able to find antibodies in you. And that's because they often are present, they are present, and they function as housekeepers, as our in-body housekeepers. They clear out the rubbish. They remove partly or partly degraded or old proteins to stop them cluttering up the system. They're often present at very low concentrations, but in an autoimmune disease, a higher amount of antibody is produced. Now, within this webinar, there are a few questions, and there are sort of multiple choice questions. So my first question for you all is, what role do most autoantibodies have in the autoimmune disease process? And I'll give you all a few moments just to ponder the answers. You have 4 possibilities. There's no role at all in the initiation or perpetuation of the autoimmune disease. The production of the autoantibody is the initiating pathological process. The autoantibodies are only produced after tissue damage by another immunological process. or they're not generally pathogenic but can perpetuate the tissue damage and inflammation in autoimmune diseases. So if you all have a little think about that for a moment, I'll give you a few seconds, and then we can think about the answer. So actually, the autoantibodies are not generally pathogenic. There's actually a handful of antibodies that do have a pathogenic role, but generally autoantibodies are produced as a result of other tissue damage, often by a cellular, a T cell and a monocyte macrophage cellular immune process within tissues. And the release of the antigens enables the autoantibodies to be produced at a higher amount. So actually, autoantibodies aren't generally pathogenic, but they do perpetuate the tissue damage and inflammation in autoimmune diseases. So the underlying pathogenic process is typically cell-mediated, and scientifically, if we were looking at it in a completely simply scientific way, we would say the best way to diagnose autoimmune diseases is by a biopsy of the affected tissue, looking for that cellular or inflammatory infiltrate. But actually, if I was a patient and somebody said, we would like to do a biopsy of your tissue or do a blood test, I think I'd probably prefer the blood test. So practically speaking, it's easier and more pleasant for the patient. Probably it's also less risky for the patient. But as that cell damage is done, the antigens are released and the B cells begin to make higher amounts of these autoantibodies than normal. And the B cells then can make antibodies to different epitopes of the antibodies, and the antibodies can be of higher affinity and avidity than is normally produced with the normal low level of autoantibodies that we have. So once those autoantibodies are made, they can bind onto the antigens, deposit in the tissue, and then perpetuate the inflammation, so the inflammation carries on. As we said, we use autoantibodies as a low-invasive way to help us diagnose and monitor autoimmune diseases. One of the very important concepts that we have to make sure we are aware of is the difference between non-pathogenic and pathogenic autoantibodies. And you remember that we said most antibodies are non-pathogenic, and a good example of that would be thyroid peroxidase antibodies. So thyroid peroxidase is the antigen. It's an intracellular antigen. So actually, for an antibody response to be made, we actually need to release those antigens from within the cell for the antibody response to be possible. And with thyroid peroxidase antibodies, the concentration is unrelated to disease activity. For the same amount of hypothyroidism, you can have one patient with very low thyroid peroxidase antibody concentrations and another with very high thyroid peroxidase antibody concentrations. These antibodies are seen very often in women, and particularly postmenopausal women. They're also seen in a variety of other diseases. For example, hyperthyroidism, as well as hypothyroidism, thyroid peroxidase antibodies are seen in hyperthyroid. And one of the good markers of an IgG pathogenic antibody is its ability to cause the disease in another person. For example, if it's crossed the placenta, IgG crosses the placenta in the last trimester of pregnancy, if the baby can get the same autoimmune disease as mum has, that's a good marker of it being a pathogenic antibody. And really, when we're thinking about thyroid peroxidase antibodies, we're looking at really whether they're present or absent. And even that doesn't have high specificity or sensitivity for hypothyroid. The other side of it is TSH receptor antibodies that are pathogenic antibodies. The antibody is directed against a cell surface antigen. The concentration is related to disease activity or disease severity. It's highly specific for the disease. And you can transfer the disease with antibodies. For example, placental transfer, somewhere between 1 and 17% of babies from mum with TSH receptor antibodies will be born with neonatal hyperthyroidism. And in pathogenic antibodies, it's the concentration of the antibody that's very important. Now, potentially we're all patients, and what do we expect of the tests that we have done on us? Well, we expect overall reliability. I think that's the easiest way of describing it. But if we put a little bit of background information, we want meaningful results. We want them to be clinically specific and sensitive. We don't want false positives and we don't want false negatives. We want the same value or concentration if the patient is stable, and that's from day to day, week to week, month to month, between labs and between methods. And particularly for a pathogenic antibody, we want to be able to detect small changes that relate to clinical disease activity, especially that reflect the condition change in the patient. And very importantly, as we're now having bigger laboratories with lots of transfer of samples, of course, patients are very mobile. Patients can go from one hospital to another. What we really want is the same result and the same interpretation wherever the patient sample is analysed. So if one sample was sent to a number of different laboratories, we would expect a comparable result. Well, are we achieving this? And I think this is a little bit of an embarrassment as a laboratory scientist that we've let this go on for so long. This is antibodies to myeloperoxidase. These are one of the anchor-associated antibodies. This is data from UK NECRAS, used with their permission, and this is the distribution in 2010. And this was antibodies to myeloperoxidase for a sample that was known to be positive for myeloperoxidase with associated clinical picture. And as you can see here, this is the range of method means in the concentration of myeloperoxidase antibodies. One of the first things to note is we've got two different units. We can have these results presented in units per mil or international units per mil. In fact, there's another international units per litre. Actually, all of these are completely arbitrary. And if it's international units per mil or international units per litre, it doesn't vary by 1,000-fold. These are arbitrary units. But more worryingly is you can see that there's 1,000-fold difference in the kind of concentrations that we're seeing over 38 methods for this one sample. Now, we in the lab know what our own method is reporting and how our own method works, but the clinicians that we're reporting these results to and the patients may not have any understanding of this massive variation in the potential result from one sample analysed by more than one different method. And the potential risk is the inappropriate interpretation of the results, inappropriate diagnosis, and inappropriate management or treatment of the patient. Now you may say, well, that's 9 years ago. Surely things have changed. Well, we still have a problem. This is data from last year. And yeah, there's a big gap in the middle where there aren't very many results, but actually we have 3 results being reported down here of very low concentrations, and then results between 80 and 90 and up to greater than 1,000 units per mil international units. So we still have this massive difference over 8 years. And I don't think it's acceptable. So we do have a problem with the kind of results that we're providing. So, my next question. What are the possible causes of such high variability in autoantibody assays? This time you have 5 options. The source of the preparation of the antigen. So, of course, any assay will have an antigen that the antibodies from the patient sample have to bind on in some way during the analytical process. So, the source and preparation of the antigen, the standardization or the lack of standardization of autoantibody tests, the differences between the affinity and the avidity of these antibodies that we're trying to measure, the very many different methods, or all of the above. So a few seconds just for you to think through these answers. If we click down, actually it's all of the above. Every single one of those components contribute to the high variability that we see in autoantibody assays. And if we look, these are the challenges or some of the challenges. So here we have the antibody variations, the variation in the antibody that the patient's producing. They may not be producing consistent antibody during the various phases of their disease. The antibodies may vary in affinity or avidity. And there's also an issue with comparability of the antibody in the patient sample with any assay standard. The antigen is another source of variation, and if we look at some of the autoantibody tests that are available, the antigen can be purified or it can be synthetic, it can be degraded, there can be lot-to-lot variation. So there are many possible sources of variation here. There are methodological variations, the type of diluent, the type of dilution, whether it's automated, manual, what the conjugate is, whether it's a capture assay, whether it's a direct assay. So we've got lots of possible variations here. And we also have what's the detection system. Are we measuring IgG and IgM, IgA? Do we have equal representation of the IgG subclass reactivity? So we have a whole range of different possible challenges. Then we have the issues like, well, it's too complicated and our lab is locked into using this particular method or analyzer. We have people say, well, does it really matter? It's okay. We understand the results. And then we have people saying, well, we use arbitrary units, because then all of the assays on our platforms will look the same with the same cutoff values. And then we have, it'll never work. We have bigger worries with glucose and TSH and point of care testing and a whole range of other issues that we as laboratorians have in the kind of tests that we do. But at the centre of the issues for autoantibodies is that there are no robust reference materials. So why is immunoassay so complicated? Well, the immunoassay for autoantibodies is kind of complex times complex. So if we look at TSH, which is a fairly simple immunoassay, TSH is one protein with two subunits. And here's a little sketch of TSH down here. If we're looking at IgG, we have 4 different proteins, 4 subclasses. We also have multiple, slightly different structural characteristics. For example, in a polyclonal response, we have a complex 3-dimensional structure. We have different stages of degradation, various half-lives. But then when you try and combine those two things, and we're trying to look for antibodies to an antigen, we then add in the complexity of the protein, the IgG, plus how the antibody is purified, the affinity, the avidity, the IgG being both an antigen and an antibody, the different proteins, the structural characteristics. So we're actually multiplying the potential complexity as soon as we start looking at autoantibodies. Immunoassay in itself is complicated. When we're looking at autoantibodies, we're making it even more complicated. So what are the sources of the antigen? Well, the antigens have multiple sources. They can be extracted from mammalian tissue, and that often involves a multitude of purification processes with heat, with cold, with salt, with alcohol, and all of those different processes can alter the structure or denature that protein. The antigen may not be entirely pure. It may be contaminated with other things that are in the same tissue preparation. And the stability of those preparations may not be either very robust or very well documented. We also have issues with lot-to-lot variation, and for some autoantigens particularly, how the relevant antigenic epitopes may be expressed. So to get over some of these issues, people have tried to make synthetic antigens that may not be necessarily identical to native proteins, either structurally or antigenically. And they may also lack the important epitopes that we're trying to look at. And there is high variability between manufacturers, certainly, but also lot-to-lot variation. As soon as you're preparing these antigens, the lot-to-lot variation can be significant. So then, once we've got the complexities of the antigen, we now have the complexities of the antigen-antibody reaction. And the binding of antigens to antibodies is variable, both in affinity and avidity. We know that some patients make high affinity antibodies that bind extremely tightly and form very stable complexes with the antigen, both in vivo and in vitro. And these high affinity antibodies are often very damaging, particularly through complement activation and destruction of tissue through complement. And they can also be quite resilient to changes in temperature and ionic strength and pH, for example. So if you've got a minor change in the buffer that you're using, it probably won't make a big difference if you've got a high affinity antibody. But if the patient's making low affinity antibodies that don't bind so tightly, you don't get the formation of stable immune complexes. Generally in vivo, they're not so damaging. But importantly, in terms of the analytical process, the complexes can be separated, can be dissociated by minor changes in temperature, ionic strength, or pH. And the very important thing to measure is to remember is the behaviour of a patient's autoantibodies is not consistent through their disease course. Often at diagnosis, the patients make a large amount of high affinity antibodies that bind very tightly and cause the tissue damage. After the patient has been treated, then the affinity of the antibodies is often considerably lower, and the epitopes that the patient may be reacting to may also change. And so one of the issues that we have is the antibodies that we're using to standardize any of these methods is unlikely to be representative of every single patient's autoantibodies. And also the quality control materials that we have is unlikely to be representative of every patient's samples. So we have many, many different issues. This is a good example of the complexity of the antigen. This is a sketch of the proteinase 3 antigen, and there are 3 important epitopes within proteinase 3. And at diagnosis, most patients with a proteinase 3 or an ANCA-associated vasculitis show reactivity to multiple parts of the proteinase 3 molecule, but particularly to these 3 important epitopes. But in remission, the reactivity to these epitopes is diminished. But during relapse, the antibody reactivity changes from the C-terminal epitopes through to the N-terminal epitopes. So some patients even show multiple changes through their disease course of which particular antigens the patient may be reacting to. Proteinase 3 is also a very interesting molecule because actually the epitopes that the patient reacts to are quite directional. So the important epitopes are on one side of the molecule, and if the antigens are coated onto the ELISA plate, for example, upside down or as a mixture, you can lose sensitivity of the assay. And these are a couple of papers that are very important in the epitope mapping and the epitope reactivity associated with proteinase 3 antibodies and ANCA vasculitis. So, the methods. All of the immunoassays have marked, they're all immunoassays, but they show marked variations. We look at the methods, we can have automated or manual ELISAs, we can have variations of ELISAs, multiplex analysis. And each one of those different methods will have different sample dilutions, different pH, different buffers. They can be captured where the antigen is bound onto the plate with a particular direction. You can have direct ELISAs, you can have methods that happen very quickly, very slowly, or you can have various detection systems. And they all contribute, all of these features contribute to this high variation of autoantibodies. Now, as we said when we started this, the IFCC Committee on Harmonisation of Autoantibody Testing decided that we were going to attempt to address some of these issues. This is a joint project between the IFCC and the Institute of Reference Materials and Measurements, which is part of the JRC, the Joint Research Council of the European Commission. And you can get information about both these very impressive organisations by looking at the websites. And we had a number of aims. One of the aims was to identify the antibodies that would be suitable for standardization, and we were particularly looking for antibodies where the concentration of the autoantibody was generally accepted to be related to disease activity, and autoantibodies where there was no reference preparation already available. And the 5 targets that we considered were antibodies to proteinase 3, myeloperoxidase, glomerular basement membrane, beta-2 glycoprotein 1, and citrulinated peptides. Now, in fact, the antibodies to citrulinated peptides is being progressed by NIBS, the National Institute of Biological Standards and Controls in the UK. But we've addressed some of the other antigens. So our first aim was to prepare and evaluate the reference materials. But also, we can't just sit and, if we make these things and they don't work very well, that really doesn't help push the science on. And so we had other things to look at, to try and pin down the sources of variation in autoantibody testing and put some sound science behind why. What are the major issues in autoantibody testing? and also, once we had the reference materials, to introduce them and assess their impact. Now, we also realised that this was unlikely to be a one-step shop. We were going to probably do one thing, start the process, and then identify other areas that may need to be addressed. But actually, this is a bit of a loop, and we had to jump into that loop somehow and start the process. And so we decided that the easiest thing, the most straightforward thing that we could do would be address robust reference materials for IgG antibodies to the antigen, but with complete understanding that this was unlikely to solve all the problems. And so the first two antibody targets, the antigen targets that we looked at were the ANCA-associated antibodies, antibodies to myeloperoxidase and proteinase 3. And very briefly, we received plasmapheresis material from a patient with antibodies to, I'll talk about myeloperoxidase here, with antibodies to myeloperoxidase and the relevant clinical findings. The plasma was converted into serum by the addition of protein sulfate. It was incubated, centrifuged, and the fibrin was removed. There was a delipidation step, dialysis against isotonic saline, then a pH adjustment, preservatives were added, then it was sterilised, filter sterilised, and then finally the antibody was transferred into 1 ml aliquots into vials in a clean room and lyophilized. And then the biggest part of this was the detailed evaluation of the reference materials that we had provided. Now, we all use the term reference materials and standards and all of those things, but actually there are some very simple characteristics of reference materials and what needs to be evaluated if we're making a reference material. So which of these are characteristics of a reference material, a certified reference material? We need to think about whether they would be homogeneous, stable, traceable, commutable, safe and ethical, whether they are available, certified, or all of the above. That's quite a list for you all to think of, so I'll give you a few seconds just to formulate your answer. Well, actually, it is all of the above. And the characteristics of a reference material and the two things that we prepared, ERMDA 476/IFCC and ERMDA 483/IFCC, these all fulfill these characteristics. And the details of this preparation is later on in the slides. But I mean, a few things to highlight. When we say stability, what we expect is the material to be stable over its lifespan and during shipment. So we have to make enough of this reference material so it provides some continuity. We also have to assess its stability, for example, in this situation, for 2 weeks during shipment and on storage at -20 and -70 degrees C. It should be, if it's a certified reference material, be related to a higher order reference material, usually a national or international reference material, through an unbroken chain of comparison, and each with a stated uncertainty. And the values for these two materials assigned in milligrams per litre, calibrated against ERMDA 470K, IFCC, which is the international reference preparation for serum proteins, including IgG. They have to be commutable, and I think the next slide, maybe I have it. Commutability. Commutability is a characteristic of a reference material to behave in a comparable way to the samples relevant to the intended use of the material. And commutability is a very important component of any kind of reference material. It needs to be available, it needs to be collected ethically. We can't just take these samples from patients. And in these situations, we actually did achieve consent from the patients for their material to be used. And finally, with all of those things assessed, we have to look at whether it's certifiable, whether those materials can be certified. Now, if we look a little bit again at the commutability, this is for myeloperoxidase antibodies, and the graph here on the left-hand side of your screen is looking at a number of different methods with the same samples, but this is using the calibrators that are generated in the kit. As you can see, there's quite a span of results. But by using a candidate reference material, we can pull those results into much closer agreement. So here we have a range between about 40 and about 210. Here we've got a range between about 100 and 120. So just by using the candidate reference material, we have a good, we've improved the agreement, good convergence of the results for 6 out of the 7 methods. And actually, for the 7th method, the results have improved considerably. But these outliers, we can't simply solve those just by recalibration, but we can make a significant impact on the results. This is also about IgG anti-myeloperoxidase antibodies, and this is the comparison of the various methods. And as you can see, the dark green is where comparison between 2 methods is very good, and there are a couple of methods that don't seem to compare with other methods. But generally speaking, this was quite a surprising and pleasing sight that we could see significant comparability between the various methods if we're using a common reference material. So, great excitement. So back in a few years ago, the first reference material to be certified was the anti-myeloperoxidase material, and the second material in February 2017 was at the anti-proteinase 3. And you'll notice, as well as the concentration, the value in milligrams per litre, we also have an uncertainty of measurement, an uncertainty of the value of here of 9 milligrams per litre in 84 milligrams and 29 in 270 milligrams per litre. So great excitement, and the values in this situation, they are not in arbitrary units, they're in SI units and with traceable values to an international reference preparation. So great excitement. But is this the whole story? Well, you can imagine that it isn't the whole story. What I want you to do is focus on the colours here and the colours going across the samples. So down here we have different samples, and across the top we have a selection of different methods. And any box that's highlighted in orange would say that the sample was positive in interpretation based on the kit insert. The yellow results are where the results are borderline, and the green results are where the results are negative. And looking at these samples, only 9 samples gave the same interpretation by all methods. And if we took out the outlying method, still only 13 samples gave the same interpretation for 10 out of the 11 methods. So although we know that we can improve things, it certainly doesn't solve all the problems. So the reference materials, however happy we can be that we've achieved reference materials, it is not going to solve all the issues. And this is some work from my own lab where we've had a number of different methods available to us, and this is for IgG anti-proteinase 3. And you can see that some samples give a very high result by one method and low results by others. But this method doesn't always give low results. For another sample, it gives a very high result and a low result by the first method. You can see that there's really quite a lot of variation in a selection of samples. So we know that some patients, or samples from some patients, do not behave consistently in all methods in terms of either the value or the positivity or negativity. So where are we now? So the first time ever, these antibodies could be reported using the same scale, and the results could be traceable to a higher order material, and the variability for a large percentage of the results should be reduced. But it will not solve all the issues of certain patients' samples behaving differently in one or a multiple of methods. Interestingly, other manipulations of the data, for example, the odds ratios, won't solve this issue either. So we have 2 things. We've got a reference material. We're very happy that it should be used, but it is not going to solve all the problems. And we have to be aware that these problems exist and be conscious that when we're reporting results, that there is some dialogue with the clinicians. So if the result doesn't fit in with a clinical picture, that maybe we should consider other methods to check that sample. So we can be very happy that this is where we've got to. And it's also interesting that when we look at the JCTLM, the Joint Committee for Traceability in Laboratory Medicine, is the international consortium that promotes this global standardization of laboratory tests, where they're very much striving towards comparability of laboratory tests. And they say that traceability values will only be possible if we have a universally recognized and accepted system, such as SI units. We also have the European In Vitro Diagnostic Directive that says the traceability of values assigned to calibrators should be through reference to higher order materials. So we have a strong steer that as laboratory scientists, we need to look at the comparability of results, reduce the comparability. Standardization is key to all of this. And in fact, there's going to be legislation that says if you're using a test where there's an international reference preparation, your test should be calibrated against that international reference prep. Many of you who have your laboratories accredited to ISO 15189, that equally states recording the metrological traceability of the calibration standard and the traceable calibration of equipment. So it says you need to know whether your test is traceable and what it's traceable to. And if there is a higher order reference material, you should be using it. Now, we've made these materials, but still I can't buy any kits for use in this laboratory where the standards used in those kits are traceable to the two reference materials that we have prepared. And I wonder, is autoimmune that special that we can sort of ignore traceability, that we can pretend that it isn't for us? I think we should all start using these reference materials, particularly, as I say, the new IVD regulation, the In Vitro Diagnostics regulation, says that if you have a material, a higher order material, you should be using it. And this will come into full application in May 2020. And in May 2022, there will be there may be penalties, there may be pressure for companies to adopt these higher order reference materials. So we have these reference materials, but we still aren't using them, even that people know about the JCTLM and the new in vitro diagnostic regulation. So what are the barriers? Because we know that we need to reduce the risk for patients, for anchor testing, for example. We need to reduce the variability of anchor testing. We need to better understand the sources of variation. But actually, we've come up against a bit of a brick wall because people say, well, it'll be expensive. It was estimated that it will cost between $500,000 and $1 million to redo all the paperwork for kit inserts, for example. It'll be complicated. People perceive that they understand what they're doing now and there's no need to change anything. The legislating bodies like the FDA, well, we need to get FDA approval and that's complicated and time-consuming. People may say, well, whatever the company does, it's really the laboratory's responsibility to educate the doctors in the variation of this test. Lots of money, no benefit, marketing won't allow it, and who knows what's right. So all of these arguments are out there, and people saying, well, we have these materials, will it really help? Now, the issue is we know that the standardization won't solve these issues that we've discussed at length today. It won't solve the variation between the patients. It won't solve variations during the disease. It won't solve the antigens that are used in the test. But we actually do have to move forward. We can't sit in our own little bubble thinking that autoantibodies are special or different and that we don't need to worry about it. As laboratory scientists, we have to do something. And there are a whole range of people, so us in this webinar who are all listening, just listening isn't acting. We actually have to start putting some pressure, particularly on the companies, on the EQA providers, maybe on the professional bodies, the service users, to educate them in the situation that we have and the potential solutions to that. Unless we can pull the results together for autoantibody tests, we are going to really lack any insight into what are the main sources of variation. We may find that patients with certain autoantibody profiles in terms of their epitopes may have more aggressive disease, or patients with a different spectrum of antibody profiles will have a less aggressive disease. But until we go through that process, we aren't going to know. We're not really going to understand what we're doing. We're just happily measuring things that maybe aren't as good as they should be. So I think what we have to do is make a very clear plea to the companies to calibrate their assays with respect, in terms of the anchors, to these two reference materials. I think initially as an option, for example, introducing a conversion factor, saying that although you measure in in units per mil or international units per litre, you can convert that into milligrams per litre. And although labs would continue to report their results in their usual units, data could then be traceable, data could be collected that's traceable to the reference materials. And ultimately, what we need is underpinning data. So we should generate robust data on the clinical performance of these assays with respect to the international reference preparations. And for example, this could populate the data required for the FDA 510K submission. We also need to encourage EQA providers to accept results in milligrams per litre, again, calibrated with respect to these international reference preparations, again, to generate more robust data. It would be very interesting if we could have EQA showing the spread of results in units per mil and international units per mil and whatever, and show whether that spread of results for an EQA sample is significantly reduced if all companies are calibrating their results with respect to the milligrams per litre and international reference reps. We, as a group of users, need to understand better the sources of variation. I've listed you all sorts of possibilities, but actually I haven't been able to put too much detail on what those sources are. Now, it is in progress, and we've done some quite extensive work with Professor Spector in the Mayo Clinic looking at the epitope mapping of the reactivity within the reference materials to myeloperoxidase and proteinase 3. And his conclusion is that the reference materials show this very broad spectrum of epitope reactivity. So it should be relevant to patient samples. But it's the patient samples that we then need to think about. How are they reacting within the method? But this will be invaluable in showing us how to proceed with antibody standardization from an evidence base, not from us, playing around in the lab saying, oh, we'll do these tests today and it doesn't matter. They may be a little bit variable, but it's fine. We understand it. We actually need, for the first time ever, to put a sound evidence base in what we're measuring and how we're measuring it. Again, supporting evidence and explanation for any FDA or CE marking. So in summary, we can certainly improve the numbers. We've made a huge advance. We now have traceable, commutable, certified reference materials for IgG, anti-myeloperoxidase, and anti-proteinase 3. We know that it cannot solve all of the problems. But without using them, we aren't going to get to that position where we can begin to understand the sources of variation and work closer to resolving those. Undoubtedly, collaborative work is necessary. The Committee on Harmonisation of Autoantibody Testing is just one element of it. The autoimmune standardisation groups, the various companies, the EQA providers, all of us in laboratories with an interest in this area, all can participate, along with the patient groups, the FDA, the CE device validation groups, professional bodies, and service users. And I'd love to say that we'd solved the problem. I mean, we certainly haven't solved the problem, but it is the first step in moving towards solving the problem of autoantibody standardization. So thank you very much.
Okay. Thank you to you, Joanna, for your standing speech, as usual. Thank you very much.
Thank you.
Now the discussion is open, and just to start the discussion, a question about, are there any other potential uses of these reference materials that you discussed?
I think one of the most compelling uses of these reference materials is trying to allow the companies to reduce lot-to-lot variation in their kit preparation, because this is a completely independent material that every company can use, but we've also been through very detailed work on its stability. So we know that if companies use this material, it will be stable. And reducing or limiting lot-to-lot variation, I think, is an important function of these materials. Although ultimately what we would like is the tests standardized against them. But lot-to-lot variation is an important issue that these could hopefully reduce.
Thank you very much. There is a question by Mike Schneider. Do you see, Joanna?
I do. You mentioned that IRM is needed in autoantibody tests for antibody correlation. Do you think autoantibody tests with only a cutoff, no reference materials necessary? Michael, I think this is an enormous problem. We have many autoantibodies that have no standardization at all. And we wanted to start somewhere and to see whether it was even possible. And I think we've proved in principle that we can make certified commutable reference materials. And I think gradually, as we become, as these materials are adopted, we can see the potential benefit of them. But also we can start looking at other autoantibodies and see whether we can use a similar process to make autoantibodies for make standards for other autoantibodies. But so far, the IFCC working group has only managed to achieve 2 certified reference materials, although there are other ones in the pipeline. So it's a very, very big process, and we are only just scratching the surface. So I don't know about. I think even with a cutoff value, a reference material would be of benefit, because at least then everybody measuring that antibody or detecting that antibody would be looking at the same kind of value. So I think it would be beneficial.
Thank you. I agree about the fact that the issue is enormous, a very big issue. Other questions? Second question. What work is being done on identify the other potential causes of variation in autoantibodies in your work, in your group?
Yeah. I think the big part of the work that we've done so far has been the work with Professor Ulrich Specks, where we've been looking at the epitope mapping. But this is really just looking at the reference material, and I know he's done considerable work on the epitope mapping of patients with the antibodies with anchor specificity. And I think this is the next area that we need to explore in greater detail, because it's possible that we may be able to define antibody reactivity more specifically. We won't just say proteinase 3 antibodies, we'll say proteinase 3 with antibody activity against particular epitopes. And if we can do that, then we may be looking at a different classification of diseases. But at the moment, without moving forward on standardization, we're struggling to even get the companies to adopt these materials and participate in this kind of development.
Thank you very much again. So, if no other question, time is running very fast. So, could you, Joanna, tell us some more about future perspective to close the webinar?
Okay. I think the big challenge that we have in the next maybe couple of years is to try and get the materials that we have made adopted by both the companies but also by us as laboratory users of the service. And that's going to be a huge piece of work, because if you think of that variation of 0 to 10 to greater than 1,000 units of a proteinase 3 result or a myeloperoxidase result, all of the laboratories are going to have to do considerable work to educate their users to make sure everybody is understanding what is happening. So this is a big challenge. We also need to think about new antigenic targets. And in the pipeline with the IFCC committee, we have antibodies to beta-2 glycoprotein 1 and antibodies to the glomerular basement membrane. And we're also just sourcing the materials to make antibodies to tissue transglutaminase, endomysium, and deamidated gliadin. So we have another a number of autoantibody targets that we're considering going through a comparable process. So they would be the next things that are on our list of subjects to address.
Thank you, Joanna. So, thank you, Joanna, again for your great presentation. And I thank very much the audience and you as a very outstanding speaker to participate to this webinar, a webinar focusing harmonization of autoimmune tests. Thank you very much again.
Thank you. Bye-bye.